Observation and comments on experimental setup. Phototropism. The response of plants towards the sunlight means phototropism. The phototropic movements are shown by shoot system of the plants. To understand about phototropism, we will observe this experiment. You can see some small seedlings growing in a pot. This pot is kept in an enclosed chamber possessing a small window only on one side. Through this window, the light can enter inside. The leaves or shoot of the seedlings are seen turning towards the window, that is, towards the source of light. Therefore, we can understand that the shoot is positively phototropic. 2. Suction due to transpiration. When the leaves of plant transpire, they create transpiration pull. This pull helps in the ascent of sap. In this experiment, we shall see this effect. You can observe the suction taking place due to transpiration. A glass tube is filled with water and a cut twig of a plant is fixed in this tube. At the other end, the capillary tube. This tube is dipped in a beaker containing mercury. When we watch this setup for some time, we can see the mercury rising in the capillary tube. This rise in the level of the mercury in the capillary tube is due to suction due to transpiration. 3. Apical dominance. To understand the phenomena of apical dominance, this experiment is set up. In this experimental setup, there are 2 potted plants. In first pot, the apical bud is intact, while in the second one, the apical bud has been removed. You can observe these plants for a few days. The plant from which the apical bud has been removed shows the growth of lateral buds. The plant in which the apical bud is intact does not show the growth of lateral buds. This inhibition is due to apical dominance. 4. Hypogeal germination. Hypogeal germination is the type of germination in which the cotyledons remain in the soil during germination. This type of germination is seen in certain dicot seeds such as gram and in all the monocot seeds. For understanding hypogeal germination, we shall use a gram seed. Upon observing the germinating gram seed, we can see following changes. The radical is seen coming out of the seed and growing downwards in the direction of gravity, forming the root. The part of the embryo axis is seen arching out and carrying only the plumule above the ground. This part is called epicotyl. The plumule develops into the shoot. The cotyledons remain below the soil. This is a characteristic of hypogeal type of germination. 5. Epigeal germination. The type of seed germination in which cotyledons are raised above the soil level is called epigeal germination. In this experiment setup, we shall observe epigeal germination. For this purpose, we shall use seed of castor. In epigeal germination, the radical is seen coming out of the seed and growing downwards, forming the root. The part of the embryo axis lying below the cotyledons is seen growing and pushing the plumule and cotyledons out of the soil. This part is called hypocotyl. The plumule develops into the shoot. The cotyledons are seen coming out of the soil during later stages of germination.